Namaste, welcome to 𝗳-CSAT program. So, as a part of Gurukula Degree College lecturers, so today we are going to discuss about the topic biotechnology and its various topics which are included in the biotechnology. So, before going to the subject, so we should be aware of the preparatory mode for the examination. So, first 𝗧𝗧 is that always we think about the subject, but before getting into the subject, to write any competitive level exam or any pattern of MCQ exam, we should have a strategy more than the subject. So, once we have the strategy, with the help of our subject, easily we can crack any type of examination where we can get into through it. So, the basic thing is that when we are writing any such type of exams like degree college lecturers, so here two different types of aspirants will be there, where some of the aspirants where they have completed their masters and Bachelor of Education and doing some jobs in teaching level. And the second category, they will be in research level, where they have done their PhDs and postdocs, where they are also still eligible to write their exams. So, based on if you see the both categories, the aspirants who are already in the teaching profession, they will be very thorough of the basics. And the other head, when we see the aspirants who are into the research and further degrees, they specialize themselves and they will streamline the subject, where here they need to emphasize on basics. So, getting into the subject reading, to my preference, I always prefer that reading a subject textbooks. The textbooks itself gives a lot of subject in it, where the pattern of textbook is also different. The reading of a textbook is also different. So, generally we read it is in a manner of gaining the subject. So, while reading any chapter or while reading any topic, so before getting into the topic, we need to look into the scientist, the contribution of scientist, the discovery of the scientist and the statements given by certain scientists. Let us see one example is that one of the scientists, he said that taxonomy, we all are aware of the taxonomy. When we perform any taxonomical studies without any phylogenetical ancestral characters, when we do not take it that consideration in the classification, a scientist says that it is something like a flesh without bone. So, such type of prominent statements are there, where they are given by some scientists. So, this information when we read the textbooks directly into the content, we may not get it. So, such type of statements given by the various scientists, that goes into the introduction part of a lesson or any topic or any chapter. So, generally we omit that topic, we overlook, and at the same time getting into the introduction part. The introduction part of textbook makes you an awareness towards that particular topic or towards that particular chapter, where it introduces and makes your mind ready to accept the content of the textbook. So, generally aspirants, they overlook this introduction part, where there are chances of asking few questions in a broad way from the introduction part, and later on we can expect one or two questions, what we call it as direct questions. When we see these direct questions, these questions will be something like asking the definitions of certain technical terms. So, definitions of technical terms that will be there in the end of the chapter, where what we call it as glossary. In the glossary, the meaning of that particular term is very well defined. So, generally these two parts, the glossary and the introduction part, generally we overlook and directly we jump into the content part, where sometimes there are the chances to lose one or two questions if we omit these topics. So, getting into the topic again, so there lies an importance that by seeing these all strategies, our textbook has lot of importance in doing this various competitive exams. So, the first basic material to study is that your textbooks. So, basics you should be very clear, and that basics you will get it from your textbooks. And these days sometimes it happens that aspirants, they directly rely on certain materials which are available in the market readily. So, those markets they will give the books which are available in the market, they will give you a direct subject knowledge, the content knowledge, but sometimes what happens is that the basics which we lose and which we get especially from the textbook. So, textbook reading is a mandate part of our preparatory mode. So, once we complete our textbook, then we can rely on the material which are available in the market for practice. Once you are thorough of your subject, and that material or the coachings where you go, that will help you a lot for the practice. So, unless you do practice, you cannot crack your competitive exams, and practice in such a way that the practice should be done with a time limit, in such a way that one question should not extract more than 1 minute. So, one question if you put it for 1 mark, 1 minute, that will give you an easy time management. If at all it is consuming little more time or sometime little less time, that time can be utilized for the other questions. So, when we concerned about our topics in biotechnology, let us talk about cell biology. So, cell biology, in the cell biology and complete the organelles, the components of this cell, they will ask the chances of asking about the cell and cell functions, the type of cells and the various types of organelles present in that. So, here sometimes at this level we may not expect such type of questions, a direct discovery questions. Let us say mitochondria is discovered by, such type of direct questions we may not expect at this level of examination, but rather little more understanding way of questions, where they will ask in a such a way the question itself is related to the 2 or 3 other questions. So, unless you know other 2 questions, you cannot attempt this. So, such type of questions are maybe chances of getting in such type of questions in the topics. So, when you talk about cell biology and the division of cells, when you take about division of cells, you have various stages and you will have various kinases where it governs the cell division parts, and also the interlinkage of the cells on whole, the working pattern of a tissue and the organization in plant and also in animals. So, this deals with the complete cell and its functions and roles and types of cells. Sometimes you may refer some noted reference books like Cooper, that is a good book and where you can refer, but I always tell you in that before going to such high end reference books, you should be thorough of your basics and the content at lower level, where that you can get it from the textbook itself. So, when we talk about again your biochemistry, biomolecules and also the various analytical techniques, when we talk about biomolecules and analytical techniques, so in biomolecules, as you all are aware that biomolecules are present in the plant cell, where they help in its all physiological activities. Apart from these biomolecules, chances of asking about the secondary metabolites, which gain lot of importance for the cell and also to the mankind. The secondary metabolites have lot of significance in it. Secondary metabolites are produced only under stress conditions of the plants, where and also they act, generally they are produced in defense mechanism. So, in such a way, apart from this biomolecules, the details and the nature of biomolecules, chances of asking about the techniques to identify presence or absence or the content of that particular biomolecule in a given sample or a given tissue or given cell. You have many techniques, and in these techniques, analytical techniques, chances of asking is that the principles, mainly generally when we are looking for this analytical techniques, generally we go on to the procedure of that particular instrument, but rather than going on to the procedure, we should be very much concerned about the principle, the working principle of that particular equipment. So, that gives you a more marks and where chances of asking the questions in that particular area. So, when we talk about again the next topic, plant biotechnology, the most happening technology that the present day, where it has gained a lot of importance. So, when we talk about plant biotechnology, so it deals with completely the new technologies, starting from the first basic procedure, that what we call it as tissue culture, to the micropropagation level. So, various media, the preparator media we will use in this biotechnology, and various techniques we use for construction of various vectors. So, when we talk about these vectors and also restriction enzymes, what we call it as RE. So, there are many universities where they are extensively working on this. So, whenever you are attempting or whenever you are reading about biotechnology, and it is mandated that the update information, where you should give little concentration on the update information, that day to day this technique is being updated with the new discoveries. So, at the same time, reading from, apart from reading from your normal textual material and this part, that is biotechnology, where you need to give stress on updated discoveries from the biotechnology. And at the same time, from the biotechnology part, so what we call it as, it is little more related to the genetic engineering. So, in this biotechnology, what we call it as, sometimes we can make a tailor made organisms, the required characters, and of course, it requires a ethical permission. With all this permission only, the techniques which we follow from this are followed and prepared, what we call it as generally GMOs. Many GMOs were introduced which have market value, and at the same time which will have less impact on our ecosystem. Then it is released into the market for the use of mankind. So, when we talk about few inventions, the latest invention, the most happening is that RNAi interference. Such type of technology gained lot of importance, where we are already aware of that flavor saver is such a variety, and many times it is asked in various competitive examinations about flavor saver. It is a variety of tomato where they have developed by using this RNAi technology, what we call it as antisense technology. So, such type of, again when we see this updated recently documented is that one of the company called Eli Lilly from US, they have developed an insulin, what we call it as humulin. So, earlier the extraction of that particular insulin that was from different organism, and now it is from the bacteria itself, the what we call it as E. coli. From the E. coli itself we can isolate this humulin and can be introduced into the human beings, where there will be the continuous production of insulin in the body and no need to take up external insulin. That is a marvelous discovery, and what we can call it as an invention in the fields of biotechnology. And at the same time, when we talk about microbiology, and microbiology is such an topic where it is a never ending topic, as we are all aware about that microbiology deals with various organisms starting from bacteria to virus. And why I call this topic as never ending is that, and day by day what we call through these mutations or through variations, various organisms are being modified themselves to withstand their external environment. And let us not say sometimes that we are sometimes becoming a different type of creatures, and we have left our food habits in a different way, where that is giving a chance for these microorganisms to modify themselves. So, such type of updated information should be gained in this biotechnology as well as microbiology. So, microbiology, chances of that is that they may ask you about the virus. Why I am talking about virus is that, as we all know that the most recently pandemic it is that coronavirus, and also our renowned institute, that is CCMB, themselves have documented 150 strains. Such a great discovery, such a great inventions and such a great research is also being done in our India, and where there we need a little more efforts to gain the updated knowledge on particular aspects. So, again when we go into the molecular biology, so in large the molecules of the cell and also present in different types of modified parts of a plants. Sometimes these plants they modify according to their external environment. So, when they modify themselves, so according to the environment or according to the need of that plant, they produce various types of molecules. So, study of such molecules and in depth study of such various types of molecules still gains lot of importance. And now presently I will introduce Dr. Somesh 𝗧𝗧 is from Department of Genetics, Osmania University, and he will explain and other topics which are going to come in your, that is a part and parcel of the syllabus for our degree college lecturers. I'll forward it to the Somesh sir. Good morning, sir. Very good morning, sir.
Good morning,
happy
Dr.
to see
Suman.
you back.
Nice to have you here. Have me here.
Yes.
So, I am happy to share my knowledge what I have been doing in the year and to prepare the aspirants for this degree college lecturers. So, basically I have been teaching biotechnology almost last 10 years. So, I did my masters from JNU New Delhi in biotechnology. So, then I did my PhD from IISc Bangalore. So, from the Bangalore I went to the US and I was there for the 4 years. I carried out the advanced research. So, after that during the selection process I joined the Osmania University. I started my own lab as a molecular medicine lab, and I have been training the PhD scholars. Already I gave a 3 PhD awarded from my research lab, and now like I have been playing a major role in the setting different competitive exams and selection process, and even for the MSc, BSc admission process also. So, I will be happy to share my knowledge what I have been doing in the university at the serving the students community as well as for the different competitive exams. So, very specifically like I have been teaching bioinformatics, molecular biology and medical biotechnology and microbiology in the university. Okay. So, based on that one, like whenever we ask for the setting up a question paper, so they will try to give a prescribed syllabus. So, no question will not be allowed to come from the prescribed syllabus. So, that is the main first criteria you have to all of you have to remember. Even though all of you know that one, what I want to convey is, so that is the, so keep prescribed syllabus in front of you and start your preparation step by step. And so, some ambiguity questions will be there. So, do not waste your time on that ambiguity questions and try to pass on like. So, we try to follow the critical thinking and evaluation when we are setting up a question paper. Some questions will be easy questions and 25% of the questions will be very easy questions will be there, 50% of the questions will be there on the to evaluate the people on the common, and 25% of tough questions will be there. So, within the prescribed time, so you have to plan that one and you have to clear the exams. Okay. So, in this one what I have been observing is like this people try to waste so much time on the tough questions, and so by end of the exams, which you know also, but still people try to waste their lot of time on the tough questions only. So, what I want to suggest the people is to concentrate on the go through the question papers, then they start answering those questions. So, when it comes to the preparation, what I want to suggest is like there are some of the standard books you have to follow in the microbiology or the biochemistry or the enzyme technology. And when it comes to the bioinformatics, when you take the standard books like David David Mount. So, those are the very tough subjects, like if you have to waste a lot of time in understanding the algorithms and other things. So, for that type of subjects you take some of the Indian version Indian editions of the books like 𝗧𝗧 is there, and that type of books will really help you to understand some basics, and later you can go for the standard versions. And as we have a ocean of syllabus is there, like when you there is no end for the preparation. So, when you start the preparation, start from the scratch and go step by step. So, that will really help the aspirants and not to get too stressed and go by the step by step. So, when you take the microbiology, you have to go prepare for the Albert Bruce, the cell and white and white, and stairs are there, Leninger book is there. So, those are some of the important textbooks. I think all of you might have been studied in the MSc, but so you have to go through those standard books, but there is no substitute for the those books. We cannot avoid those things, and you have to step by step you have to further move on to the other subjects also. So, basically bioinformatics, some questions will be there, like almost 5 to 10 questions will be there in the exam, and we also have biostatistics questions will be there, and biochemistry questions will be there. So, keep your syllabus and no question will come out of the syllabus. That is a for standard known, like those are the instructions we will try to give it to us from the convener. So, no question should come from the prescribed syllabus. So, what I suggest is strongly keep your syllabus and start preparation and follow the standard textbooks. So, that will really help all the aspirants to have ease of that one. Without the stress, you can move further and prepare that one, and you can prepare step by step. Okay. Yes. So, I will go into the in depth, like yes. Suman will share.
So, what I mean to say, actually before going to such a well versed reference books, what I suggested what I suggest to the aspirants is that the basic material should be our textbooks.
Yes.
So, that gives lot of basics to them, which gives sometimes with chances of asking 2 to 3 questions from the basics itself. So, yes. The 2 different criteria of aspirants are there in the degree college lectures. So, in the same way, what I mean to say is the subjects in different genetic engineering and biostats, yes. and you are aware in that and you are well versed in such subjects.
Exactly.
What sort of questions we can expect in such a genetic engineering, and as you know that genetic engineering is a never ending topic.
Yes.
So, every day is a new challenge in the genetic engineering. And the same time, the genetic engineering has to be supported by this biostatistics.
Yes.
For its novelty and its proving. So, what type of questions we can expect in such type of topics, sir?
it's like when we started to keep the questions, like when we start setting up the question papers, what we try to consider is, so like each version we try to take the easy question, one tough question from the each unit.
Yes.
Okay, so and one average question, so that way 3 questions should be there. Like, so in that one, some easy questions like what is the Z score we try to calculate, what is the bit score, what is the probability of this one, or the what is E value, and we have a different techniques are there when it comes to the bioinformatics, what is the blast. So those things you had to learn, and what is the nucleotide database are there, protein databases will be there, what are the different tools we try to use in the bioinformatics for the data analysis, and chi-square values, and test of hypothesis will be there, and normal distribution. So from those things only we try to ask some easy questions as well as some of the applied questions. So if you find some tough questions, not to waste in that one, but when you are preparing, so step by step you have to prepare from the scratch to the higher end too.
Yes.
Yeah. And when it comes to the other subjects, like we have a genetic engineering, or the in the all of us know that one, like we have a restriction enzymes will be there from that one. So what are the contemporary topics, also one of the 2 questions we will try to ask, what is CRISPR-Cas9, or the what is the application of that one, and how it started, what are the restriction enzymes, how do we do the traditional cloning, and how do we ligate those things, what are the T4 ligases there, what are the DNA damaging events are there, what are the mechanistics are there, and what enzymes they play a role in this one, how this is like, as we told in the nature, whatever the resources we want, we have abundantly resources are there, but we had to people want to have a selections will be there, like some people they try to, we want to have a seedless grapes are there, all those things we have been. Biotechnology is not wavered, like everybody people will be enjoying the biotechnology products, like what we eat, watermelons, seedless watermelons or seedless grapes. But how do we produce that one? We had to analyze the genes which are playing a vital role for the contributing for the seed formation, and we had to knock out those genes by analyzing the what are the group of genes, like some each trait or the local trait is contributed with the sometimes single gene, sometimes multiple genes will be there. We had to identify that one. So this bioinformatics plays a major role in the physical mapping, and so when we know the accurate location of that particular genes, so then we give it to the that information to the breeders. So then they will go and try to produce that one. And even same thing also, like even in the drug designing, other things also, we have some of the standard tools will be there, and so what gene is playing a major role, like what are the drug we try to design in the biotechnology, the pharma industry. So those for the, as we are evolving, even pathogen organisms also evolving. So this is like evolution is not only the our birth, right? Even pathogenic organisms also keep on evolving. Okay, so as part of that one, they try to acquire some genes, and what are the novel genes are there, and as part of my research, what we try to do it is like even the across the globe, those genes should be essential for the survival of that pathogen, and those genes should not be there in the host human organisms. So then how do we carry out? So then we have to do the genome wide analysis, and we have to identify those particular genes, and how do we identify this one? So that type of questions, like so we need to have a sound knowledge in the particular field. So that's what we try to test it, and prescribe technologies will be there. We also have a once some enzyme is identified, we had to upregulate this expression or the purification of that one. So when you go to the bioprocessing engineering, so we had to be that process should be viable. So this all the commercial people, when they are putting up, like we have insulin is there, like when the people try to produce in the humulin, so is it the viable, or the is it can be withstand in the commercially. So where the downstream processing and all those things we had to check that one, and we had to from the pilot scale to the upgradation. So what type of things we had to consider that one. So based on the syllabus, the biotechnology step by step we kept it up. So we have that syllabus completely. So when you see this one, we also have enzyme technologies there, or environmental biotechnology is there, and so in that environmental biotechnology we try to look for the gems, so which are means genetically engineered microbes. So we try to engineer those microorganisms to produce the high yielding products what human is required, or the to clean up the oil spillage in the oceans. Even for that one, we try to do these genetically modified organisms. So, being a biotechnologist people, how do you produce those things, and what type of technology we try to use it. And when it comes to the even biochemistry also, we try to use the Wilson and Walker is there, like if you prepare that one. So, basic analytical techniques you can get it from the Wilson and Walker biophysical techniques. So, where they try to explain from the pH setting up and what are the how do we calculate the small buffers, and many things will be there, and SDS is there, and gel electrophoresis, column chromatography. Even from that one, from the techniques also, as part of the biotechnology, we try to ask the 8 to 10 questions on the technical basis also, analytical techniques also. So, we should not leave out those techniques also. This is a biotechnology, means everywhere technology involves. So, what are the regular techniques we try to use it, and we try to ask the 2 to 3 questions on the analytical part and analytical techniques also. So, you cannot leave up the left out the any topics which are prescribed in the syllabus, and you also have to have a sound basic knowledge. So, even so as we told, so we try to ask some basic questions. The people who are reading high end, so they try to forget about the basic understandings. They try to get confused with the basic questions, where the basic questions we try to give the options. When we give the options A, B, C, D, so sometimes A and B almost relevant answers will be there. So, in the MCQs what we test is what is the most appropriate technique. So, we have to select that one. So, like when you try to see the application of any ion exchange, or the size exclusion column chromatography, or the affinity column chromatography. So, what type of things we try to use it in the affinity column chromatography. So, we try to use the nickel NTA. So, that is the we try to have a his tag, and so we try to attach that one. So, what are the methods we try to use it? Like we try to give the questions in the purifications. We do use the copper and other things also, but most precisely we try to use the nickel NTA, this thing. So, when we try to see the magnesium and even the copper, we try to put it up. So, when you have a nickel answer, you are supposed to pick the nickel. Why? Because that is the tag is there, that is the most regularly used technique. So, you have to choose that one and not to get confused with the other thing.
So, that is what actually I was telling earlier. So, in the competitive level exam, generally the knowing an answer will be very easy. But understanding the question will be little difficult and higher end. So, aspirants need to have a little in their mind is that not something like doing a rote memory or something like it is learning a subject. More than that, they should understand the subject, what they are reading it. So, when they understand the subject, that understanding will definitely help in solving any type of questions in the examination hall. So, where knowing an answer will be very easy, but once we understand the question, that will be very easy to answer it, what I mean, sir.
Exactly. So, they had to understand the questions, like sometimes even though they know the answer, they try to get confused, like so that is why they had to spend some quality time on the reading the question paper, and some when you understand the question appropriately, that is answers are before you only. So, so people they try to get confused with that one. So, not to get confused, so those are the basic things, but still you have to follow those things that really try to give a edge over the other people. So, when you selecting people, and for you there is hardly 2 to 3 marks difference will be there, even the ranks will try to change up drastically. Okay. So, those questions are very have to focus and try to understand those things, like even though you know those concepts, you try to get confused. Why? Because when you are setting up a question paper, we try to follow the Bloom's methodology, like there will be critical thinking and evaluating how much understood the subject. So, we try to keep the some set of questions for the to evaluate your critical thinking, your analysis power, and your memory. So, absolutely you have to have a hard work and memory also this, but even analytical critical thinking when it comes up. So, even those things we try to check that some questions will be there where you have to spend some time and you try to analyze. And when you are setting question paper also, we try to see that those questions you could understand it within the given time. Okay, so those things also we try to follow, like when you are setting up questions, we prepare more than what you are preparing also, that you should not think that there will be always tough questions or the easy questions or this one. So, this is always is a relative grading. So, last time how many marks we got it based on that one will not be depending on that criteria, but every set like it's a relative selection will be there. So, where is your expertise is there, try to go into that one, try to read those things. And so, all the contemporary techniques also, what are the advancements of these techniques when you see this syllabus like in the enzyme technology or the bioinformatics.
Sir, at the same time, when we see the syllabus, there is a topic bioinformatics.
Yes.
So, it is an most recently and most happening specialization
Yes.
in the sciences, where it deals with proteomics and genomics and other computer knowledge. So, when we see that the students who are coming from the basic sciences, to my knowledge they will have little awareness towards this bioinformatics. Are there any prescribed books, or is there any something like a textbook, something like that in the syllabus where they can understand and get through these topics, sir?
Yeah, that's what in the starting of my session as I told, bioinformatics, when you go for the standard books, they it will be very hard to follow them. So, go for the basic books, like as I told 𝗧𝗦𝗔𝗧, applications of bioinformatics 𝗧𝗧, that is a very simplified version they have written. And so, most of the questions we try to get it from the this one, like from the 𝗧𝗧 book, and one or two questions will be coming from the 𝗧𝗧 book, because that try to cover the most of the basics of bioinformatics, like nucleotide databases and protein databases, and as well as the metabolic pathways and proteomics and genomics. And we also have a Needleman-Wunsch algorithm, Waterman-Smith algorithms, BLAST and FASTA is there, as well as we also have some of the matrices are there, okay, PAM matrices, Blossom matrices. All these things are very simplified version in the Indian language has been written. So, same things will be there in the David Mount and other books also, 𝗧𝗦𝗔𝗧, but those are little tough to follow for the beginners. Okay, so if you are already have a well versed, or if you are carried out a 1-year course in the bioinformatics, a 6 months course in this one, so you can go for the higher end. But similar information we could get it into the some of the basics books also, which you follow from the 𝗧𝗦𝗔𝗧, or the some of the 𝗹𝗶 series is there. So, even that one, or the online easy biology, some tutorials are there on the internet also. If you have some topic, if you are not understanding, so this is like a YouTube, we can get it the lot of information, you can just give one click away. So, if you click on that one, they precisely try to explain step by step that one. And even when I am preparing for the some of the topics in the for the classes, I try to go to the YouTube, and I do take the help of the YouTube and internet material, and what are the research topics which are published in the recently. Even though I study publishing like a teaching for the almost 10 years in the university, before going for the class I do spend for 1 hour what are the techniques are happening, what is the new things are coming up, how do we present the students, and this way. So, we had to collect the, like we have all the material is available on the internet, but what to take, what not to take, and so that will, you should not spend the too much time, like you have to make a for the unit wise and try to prepare that one, and by that you have to cover the whole syllabus. So, do not some people what they try to do it is they try to completely avoid the biostatistics or the bioinformatics. That is not at all a good idea as per my knowledge. Why? Because as I told, there will be easy questions from the biostatistics or the bioinformatics or the enzyme technology or the bioprocessing engineering. So, you will be missing those simple things also, like first of all you prepare the basic concepts of the all the subjects. So, then go for the your topic of interest, and where you try to cover the hard questions also. That is what I suggest, and that is what is I encourage the students to follow the this particular aspect.
Yes.
Okay, so, this
and
is
at the same time, sir, while preparing there is a technique called TTR, developing a TTR. TTR stands for tips to remember.
Okay.
A small example in our subject is that BAD, B-A-D. So, this gives an information about D is for dikaryotic stage, which is shown by basidomycetes and ascomycetes. So, developing such TTR by the students of its own choice, that will help them a lot where to answer the direct questions.
Yes.
That will reduce an high time. That one question 1 minute means hardly they can answer this such type of questions with the help of TTRs. Within 10 to 15 seconds they can attempt such question.
Yes.
And the remaining time easily they can use for some other questions.
Yes, exactly. So, I think all these are the lecturers who are going to teach for the MSc. So, they will be knowing this type of tips, and they will be they are well versed. So, they do that is a good suggestion actually, it's in fact. So, they can have that short tips where they can memorize this one. So, this is the way we had to clear the exams, like everybody they have their own ways and means of preparation, but yeah. So, this type of tips really help the aspirants. And when it comes to the textbooks, like for the molecular biology or the biochemistry, try to follow the standard textbooks. Okay. So, but when as I told, like for the biostatistics or the bioprocessing engineering, so if you go for the standard books which are from the foreign authors, you'll be getting confused, and you had to spend quite some time on that one, and you will be exhausting over there only. So, for that one, like what I said, there is a number of series of books and tutorials are coming up. Okay. So, if you have a very short time is there, so when I am preparing for the entrance this one, so some people are there, Viva series, one of the really helped the students, like within the short time they can get a precisely they can get a concept of the some of the topics. So, that is available in the most of the libraries, and even in the bookshops also you can get those things, and they can get prepared also. Okay. So, sometimes even though you know that one some of the answers, you will be getting confused. So, statistically follow the protocol, like we have a transcription, translation, and post transcription modifications, and people also now we have in the part of the syllabus we also have epigenetics is there, DNA methylation, histone modifications. So, many things also there. So, we can prepare one after other one. So, biology is always exciting. So, which is not simple but very complex as we go into the in depth and depth. So, it is like we try to understand how these cells are getting coordinated, how these proteins are coordinating. So, like when you see into the biology, so from the birth to the death we will have same DNA will be there, and but with the we have a regulatory systems are there. So, like if you try to understand the holistically, so it will try to really help you, and try to memorize will be very easy. So, how this DNA is expressing, what type of modifications are happening, and even though we have same DNA is there in the all the cells, so why some genes are expressed, like when you have small baby some genes will be expressing, but when you reach into the teenage some secondary sexual organs and other proteins will be expressing. Okay. So, how this is happening, like when you see into the cells also we have when the Human Genome Project is finished, so they have hardly 18 to 22,000 unique genes are there, but they try to produce almost 1,50,000 proteins are there. How this process is happening? So, like if you prepare that one, you try to what type of mechanistics are there, and when you see into the particular gene structure also in the we have upstream elements, downstream elements, and we have most of the genes will start with the AUG. So, we try to see the like a molecular biologist or this biotechnology people, what are the things which are happening in the regular basis, like when you see into the water always flows down only. So, what type of phenomena is there? So, same thing. So, as per the science we always believe that evolution happens step by step, we do not believe in the any godly creation or other thing. So, science is completely against that one. So, when you keep those concepts into that one, how this is happening. So, in the holistic approach if you provide, so you will get fascinated about that one, even tough topics you can remember precisely. Okay. So, how this regulation is happening, like a same DNA will be there from the zygote from the birth and to the your deathbed also, but during the process we have even how this is happening. Okay. So, even when you see into the nature also, like a lot of things are happening. So, when you see the take the virus is the connecting link between the living and non-living. So, then it will try to get fascinated about that one. Okay. So, what type of proteins are there, why how this is could be like when outside the living organisms becomes like a sand particles, but for the metabolic purposes or for the replication it has to be there in the living organisms, and that way when you know the concept you can remember the much more difficult topics also. Okay. So, that is what I try to tell the students. So, get fascinated about the biology or these concepts, and so these things we very easily can remember.
Yes.
That is what it says.
And at the same time always I surprised to hear the words of the students coming after from the examination hall is that paper is tough or something like completely unrelated questions were being asked. So what I mean to say, the purpose of the paper setters is that to know the understanding level of an aspirant. It is not something to make them or throw them in the difficulties. So
exactly.
what is your
this
suggestion,
is
sir, on that?
No question paper is as I told when we are from the convener of this any examination, this one. So they try to give a very strict instructions. No question should come from the out of the syllabus, and so the so we will not be having any question outside the syllabus, and so that is like this test is to screen this one. We are not evaluating that one. Say among the bunch of the people, who is the best one. So no, as I told, all questions will not be tough, all questions will not be easy, all questions. So not the this end or that end, like some 50% questions will be there to test the average people, some 25% questions will be there from the simple. And so these people, that's what, like when they are trying to attempt, so these people try to focus on the those 25% of the questions which are the tough, and they will be wasting lot of time, and they will try to find that one only. So first of all, what I suggest these people when they are preparing in the preparation or in the exam also, so try to give a quality time reading the question paper. So then you go ahead, even in the preparation also, don't leave the any unit, all units prepare that one step by step by starts. So this is not my cup of tea. Some people will think that one, they will try to keep aside. That mistake you should never do that one. So take the syllabus and try to prepare that one. So with your minimum eligibility is there. So like if you have 55% marks is there, so that means you have a, of course you will have a basic knowledge will be there. Like if you spend some time on the biostatistics or the bioprocessing engineering or the enzyme technology, of course you can get a marks in the those simple questions. Okay, so but if you completely avoid that type of questions, it's very hard to get selected. So even though question paper is tough for this one, so that is not the evaluation criteria. We are just doing the screening. So who is the best from this one? So if it is a question paper is tough, means that will be same thing will be tough for the others also. Okay, so don't worry about the top things, like what is the best. Like sometimes cutoff mark will be there 75%, like 40%, sometimes will be there 50%. So based on the how much people are scoring, so we try to keep the cutoff. Okay.
At the same time, sir, related to the subject, so when we talk about this updated information, so an aspirant always cannot rely on the internet information, and at the same time availability of the internet is not always equal to all the aspirants. Some are from the rural areas and some are from urban areas. Are there any magazines, something like that, where we can subscribe that with the updated information, or else are there any websites there where we can search for latest updates in the science and technology, sir?
There is a number of journals are coming up, and there is a like in the market they are coming up. But what I suggest is like this publication, even in the internet also, sometimes we do when we set the question paper, so they find that this answer is correct. So don't rely on the completely on the internet resources. Ultimately you have to go back to the standard textbooks, read that one for the to attempt the MCQs. Like when we set up this question papers, so they tell that in this internet X and Y website, so this answer has been given, so that should be the correct. Okay, so they will try to claim that one. That is not the we don't rely on the internet resources. Like internet resources, anybody can put it up, anybody can make a YouTube video and they can present, but that will not will be not for the subject expert. We always rely on the standard textbooks and what is the answer is given, and so those things we have to follow up that one. Okay, so even all of you have to prepare for the Michaelis-Menten equations and some of the tough things which are there will be short questions will be there, this type of things. Okay, so what is molarity, what is normality, even in the buffers preparation, what is buffer, and even in the enzymes production, like Yeast-Baker's is there. So like what is the pH where we try to use it, like 4 to 5 will be there. So there will be sometimes to get confused people, they will try to give the 3 to 4, 11 to 12, 8 to 9. So but so people will think that there is no answer from the 4 to 5. So should I go for this one or not? So we have a 3 to 4 is there. So in that cases you have to choose the 3 to 4. So what is the most appropriate answer is found in this one. Okay, there will not be any completely negative question. There will be answer is there. So you have a question, there is a answer is there. So how do you choose that one? How do you do the analysis? That part really matters, and there it will give the edge over the other people. So, I strongly, as based on my experience, I try to tell you that spend a quality time on the reading the prescribed syllabus and even during the process of your preparation, and do not leave out the any topic which is prescribed in the syllabus. So, even the prescribing syllabus also we brainstorm up lot of things and we have a lot of thought process will be there. So, this topic should be kept in the enzyme technology, this topic should be there in the genetic engineering or the microbiology. So, based on that one, so those topics will be selected. Okay. So, based on that one, so even we spend a quite some time and prescribing syllabus even in the setting up the question paper, and so even same thing you have to take your time and to prepare the each topic. So, there some topics like even the microbiology, it is like a question, there is a many topics will be there, but all the topics will not be keeping in the prescribed syllabus. Okay. So, that is why do not read the from the first page to the last page. So, be smart and try to see the syllabus, based on that one try to prepare the this one. Okay. Yes. So, preparation should start from the prescribed syllabus. Yes. That is what I strongly say.
Yes, yes. And at the same time, sir, students sometimes they ask me about the weightage of the topic or weightage of the various headings. I surprised to hear that, sir, what is the weightage or what is the importance of that topic. Then I say that syllabus is framed in such a way that every topic is important. That is the reason they kept in the syllabus.
Yes.
So, weightage cannot be predicted. So, weightage is such that already appeared questions based on the topics, the weightage is presented to the students or presented to the mass media.
Yes. So, yeah, that is a good question. So, like what I want to tell is like when you are setting up any question paper. Yes. So, because I have been setting up this one. So, they try to convener will try to tell us that each unit minimum we try to give the 8 to 10 questions, or like if you feel the sometime important we can give the other thing, but we try to follow those instructions for each unit. We try to give the 8 to 10 questions. Okay. So, if sometimes we do not like if we feel that microbiology, we are testing for the microbiology and biochemistry is there. So, we may give 12 questions, 8 or like 1 or 2 plus or minus 2 questions will be giving, but in will not be breaking the instructions as such that. So, there will be questions will be there, 8 questions will be there from the biostatistics, and minimum 8 questions will be there in the bioinformatics, and 8 questions will be there in the enzyme technology or the plant biotechnology. So, like if you are missing 2 questions here and there, so in that plus 2 questions will be coming from the some of the important topics, but there will not be all questions will not be coming from the one unit. So, that will be following very strictly. Okay. So, that is the we try to keep it in the mind, and will those are the things makes us to set the question papers with those instructions only. We will try to follow that one. Okay. So, enzyme technology and Wilson and Walker also very one of the good book for the analytical techniques. So, the and Steyer's book is there, Leninger is there, and this cell 𝗹𝗵𝗺𝗺 is there. So, that is what I used to read. And so, for the microbiology epigenetics, the gene book, so that is also very well versed, and they are precisely explains the transcription, translation, epigenetic modifications and all the topics. They have a beautiful pictures will be there. Okay. In the big books, what is the advantage is, so each figure will speak up almost 1000 words. So, when you go through that picture, so that will try to convey the much more easily. So, that way, so you spending lot of time in the verbose instead of that one, try to go to the picture when you are preparing also if you are textbook. So, try to see the what type of protocols are there. If you have step by step, they try to flow charts will be there, try to see the flow charts and pictures you try to understand. So, then if you are not understanding, go for the inside of that one. Okay. Inside also is necessary, but pictures will try to give the very precise, like when you are preparing also try to make a one running notes. So, these are the topics I covered today, and when you go back and see this one, you can precisely can recall, and you can that will really will help you. So, not only reading, and you have to have a notebook also, you have to write down the key points, and when you are when you are going for the exam in the one go, you can in the overnight you can go through the all the key points, and you can cover the complete syllabus also. Okay. So, writing is like a writing in the notebook is equivalent to 21 times your reading. So, when you write this one, you need to have you use your brain and hand and everything, that will be coordination. Okay. So, whereas when you are just reading, only eyes and brain will be. So, but here you need to have a complete coordination. So, what are the points you read, make it up into the running notes and put it up this one, and follow that principle. Okay. So, that will really help you to understand the topics in the very easily. Yeah.
At the same time, group discussion also, it makes a lot of percentage, up to 80%. According to the psychology, it says that 80% of grasping occurs in the group discussions.
Yeah, of course, everybody will have their way, one way of understanding each topic, like when you put forward, when you put into the discussion. So, that is the place which brings the difference between the state universities or the some of the private colleges, and when it comes to the campus students. So, they do get a different platforms to discuss the topics, and so group discussion absolutely helps in the preparation. So, you can discuss with these things with the other colleagues who are preparing in that one, and that will really help each person to understand their perspective as well as your perspective also. Okay. So, that will really help all the aspirants who are preparing. Okay. So, they are you try to, what I suggest is, like, you try to improvise, like people when they group discussions also, people they try to go into the, so he knows more than this one. So, they try to give it up then and there only. So, do not compare with the other aspirants, try to improvise your knowledge by compete with yourself. That is what I will try to suggest my students also, do not compete with the others. Okay. So, these standard books really help the aspirants, and why? Because they have a standard descriptions will be there, there will not be any misleading will not be there. When you see the here typo errors will be there, textbooks are proofread, there won't be even spelling mistakes also will not be there in this this cell or the gene book or the Leninger stairs, very hard to find. But when you go to the local publishers, they will have many mistakes will be there, even concepts will be there, they try to explain in their own way. Sometimes core of that part will be missing this local publishers. I am not demeaning that one. So, even we do have as you told, subject experts are there, we are competing with this one. So, but when so that errors will come due to the typo error or the proofreading everywhere. So, these are the books across the globe, they are proofread and they are scrutinized. So, I suggest for the basic books like microbiology, biochemistry. So, like we have microbiology is there, and enzyme technology is there, bioprocessing engineering is there. So, we have many books are there, like when you go to the so when we prescribe the reference books. So, we try to give some of the so each unit wise I have some of the reference books as I prepared, but I could not bring it to here, like we will try to. So, each topic wise we have some of the reference books which can be, you guys can prepare very well and can follow that one. So, I prepared some of the material step by step. So, we will try to share.
It was a good discussion, sir, and at the same time awareness to the students for the preparatory of exams. And after this one more session will be there, which is dealt by other physics pedagogy, and these are the tips where you can take as a notion for preparation in the subject called biotechnology. Thank you.
Yeah, I thank you for this opportunity for the TSAT and other members. I hope our expertise and my explanations will really help the aspirants to prepare and use this information for their benefit. Thank you.